Hi everybody, this is your second video on experiments. In this video, we are going to talk about the language of experiments, principles of experimental design. We're going to talk about blindness and statistical significance. So a big bulk here in this video. All right, so let's go ahead and jump into it. All right, so in the previous video, I talked a little bit about treatments. So I want to give you an explicit definition here for what a treatment is. Okay, so a treatment is a specific condition applied to the individuals in an experiment. Okay, so it's the different levels almost of what you are imposing on individuals, what you are deciding to change as the scientist. Okay, there may be certain times when you want to test more than one variable at a time. So, and like design an experiment around more than one explanatory variable at a time. So we can use the plants example that I used in the last video. So say you wanted to actually do that experiment well, but you wanted to test both fertilizer, like types of fertilizer, and also like a high temperature room and a lower temperature room, right? So there's multiple explanatory variables there. There's the fertilizer content or the type of fertilizer, that's one, and the temperature, that's two. So your treatments would be a combination of the specific values of those variables. So the best way that I can kind of describe this is if you think about your variables as like a table where, say, we had like high temperature and low temperature, and then we had no fertilizer, fertilizer 1, and fertilizer 2. Okay, so there's 6 total treatments here, right? These would be your 6 treatments, right? High temperature, no fertilizer, high temperature, fertilizer 1, high temperature, fertilizer number 2, etc., right? So this is what I mean by multiply the number of levels for each explanatory variable of the experiment, okay? Another term that you'll hear or see a lot is this term experimental units, okay, which is essentially the individuals or the subjects that you're working on. So it's just the overarching term for that. So the exact definition is it's the smallest collection of, if I could spell, that would be awesome, of individuals to which treatments are applied. Okay, and when those units are human beings, we call them subjects rather than experimental units. Okay, so when you're talking about experimental units, you have to think about, okay, what am I actually applying the treatment to? Am I applying it to an individual person? Am I applying it to a classroom full of people? Am I applying it to one box of some product that was made, right, etc. Okay. All right, so let's talk about the principles of experimental design. So the design that you see on the left-hand side of your screen is a pretty basic one, right? Like a very basic one, right? Where you start with your experimental units, and then you apply a treatment to them, and then you measure the response, right? That works really well in a lab, like really, really well in a lab, because you have all of these controls, right? But that doesn't work out so well when you are dealing with actual like people, or you're working in the real world. Okay, things can happen, right? A lot of the times we have biases that show up, right? Not maybe that's anyone's fault, right? It's just we live in the real world and bias exists in the real world. Okay. So the way that we combat bias is by using random assignment. Okay. So what we do is we take our group of experimental units and then we assign them to one of our many treatments by random chance. Okay. So they don't have choice in which treatment they get, right? Because there could be a bias there, right? We try not to give those who are putting them in groups a choice either, right? Because they might have some underlying bias, right? So we always want to use random chance to hopefully eliminate all or as much bias as we can. Okay. And you would use the same process to do that as the process that we used when we were looking at this with observational studies, right? Using either a random number generator or pulling strips of paper from a hat, right? So if you're asked to describe how you would do that, it's exactly the same process, right? So you would, number the experimental units from 1 to n, where n is the sample size, right? And then use a random number generator to generate, say we had 20 subjects in a group. So you generate 10 random numbers, give them the first treatment, and then the other 10 random numbers would be given the second treatment, for example. Okay. And we use the random assignment to try to make the groups as even as we can, right? To take the experimental units and put them in groups for the different treatments as evenly as possible. In the real world, sometimes that doesn't work out. As long as they're like as close as they can be, that's good. All right. Okay. So the random assignment is one of the 4 principles of experimental design. Okay, so you have to have these 4 principles in any experiment. Okay, so the first is comparison, right? So there's no point to do an experiment if you're not comparing one treatment with another, right? Because you can't establish a cause and effect relationship that way. Most of the time, you're trying to figure out, okay, does me changing this one variable, maybe at different levels, affect the outcome, right? Or the response of my subjects. So you have to be comparing 2 or more treatments. Okay. Random assignment, I already talked about, but that's when you use chance. So the hat method, random number generator, etc., to assign experimental units to treatments, right? And I talked about this as well, that doing this creates roughly equivalent groups, right? And one of the reasons that we do this is so that we can balance the effects of other variables among the treatment groups. So remember when in the previous video I was talking about confounding variables, right? You do as best you can to control and eliminate those confounding variables, but in the real world, it's almost impossible to eliminate them all, right? So using random chance and the natural variation of the universe, right, that we try to balance the effects of those other variables. Okay. Principle number 3 is control. Okay, so we want to keep the other variables that might affect the response the same for all the groups. So if you think back to the previous video when the example that one of the examples that we did was on battery life and screen brightness, right? So remember in that problem, they used the same brand of laptop. All the laptops were new, etc., right? So we were keeping other variables that could affect the response the same, right? It's not necessary that you have what's called a control group where you apply no treatments. You can if you want to, but it's not necessary in every experiment, okay? Control here is referencing, it's not a noun, it's a verb. It's you are trying to control as many of the other variables that might affect your outcome as possible. Okay. And then the 4th principle is replication, right? If I just have 2 subjects, I assign one to one group and one to the other, and then I measure their responses, that doesn't do me much good because, again, it's very possible that something else affected how they responded to that. So I need more data, right? I need more subjects, more experimental units so that I can really make a definitive statement that if I see a change or a difference between the 2, 3, 4, however many treatments you have, that change is due to those treatments and not to some other variable, right? So the point is that we're trying to distinguish the differences in the effects of the treatments from just any chance differences that show up between groups, right? We live in the real world, and the world is full of natural variation, right? That's how the world is. So when you're conducting an experiment, you have to know that the changes that you see are not because of that random natural variation in the universe, or the chance differences between those two groups, but rather because of the differences that you imposed as the scientist. All right. So we have this scenario here. Okay. So this study or this experiment that happened. I'm going to pause the video here. I would like you to do so as well. Read this over and see if you can pick out the 4 principles of experimental design in this scenario, right? So I'm going to pause and I'll see you in just a second to check your answers. All right, folks, we're back. So let's check these answers over. I highlighted some important things in the scenario for you to pick out. These were the things that kind of gave away those 4 principles of experimental design. So for comparison, the design of the study was to compare students who were multitasking during a lecture versus those who were not multitasking. Okay, so you're comparing 2 treatments. For random assignment, they stated it directly in the scenario that the researchers shuffled the instructions and handed them out at random. Okay, for control, there are lots of ways that they controlled this study, that they went to the same university, all of the students went to the same lecture, they all had to take notes on laptops versus, some people writing with paper and pencil. There was none of that. They all had to take notes on laptops, and they all took the same comprehension test of the lecture at the end. Okay, so they controlled for a lot of possible confounding there, right? And then for replication, there were not 1, not 2, not 3, but 20 students in each treatment group. So that's enough replication that the differences that they detected, we could say, are due to the multitasking rather than chance variation due to the random assignment. Okay, so give me a second. I need to clear my screen, but after that, we're going to come back and I'm going to talk to you about completely randomized designs and blindness, and then a couple of other things. All right, y'all. So the most basic and really the most common type of experiment is a completely randomized design, right? And it's exactly what it sounds like. You take your 2 treatments and you assign your experimental units at random to those treatments, and then you conduct your experiment and then pull all of your data together at the end to compare the results. Okay, some of these experiments can have a control group that receives what's called an inactive treatment. So I'm not going to draw it on the right-hand side because it took me a little while to draw that. I can freehand it a little bit better here on the left-hand side. So rather than just having group 1 and group 2, there could be a third, group 3, where they don't have a treatment, but they would have no treatment. Right? That's your control group. Okay. Again, reminder, your control group does not mean how you control an experiment. Like, they're 2 different things. The control group and the control principle of experimental design are different. Okay. Right. So, here, one of the key things that I want to point out to you is if you are asked to design an experiment on the AP test. You do not have to write out everything. You can draw a diagram like this, and it counts as long as you do a couple of things. First, you have to identify your sample size for each group. That's what this N value is right here. Okay, and when you have random assignment, you have to describe your random assignment process. So that's your steps, right? The one, number subjects from 1 to N. 2, use a random number generator, ... right, to select however many unique subjects to go to group 1, all others go to group 2, yada, 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 etc. Okay? And then after that, as long as you have all those things, you don't have to write things out like in full paragraphs. If you're more of a visual person and you like to draw things like this, you can. You can also write these things out in bullet points if you want. Okay, for stats, you don't have to write like full paragraphs. Most of the time, bullet points will suffice. They're not worried about how well you can write. Not really. They want to know that you know the statistics. All right. So this is what a completely randomized design looks like. It's just where these say experimental units and group and treatment, you would have to provide some context, right? Whatever the context of the study was. So what are the experimental units? Group 1 is what? Group 2 is what? Which treatment, like what are the treatments, right? You just have to make sure that you talk about those things in context, right? And as long as you do, you're golden. All right? So let's talk about blindness. Okay, this is something that you may have heard before. You may have heard the term double blind before. Okay, but this is really where it matters and where it counts in statistics. So experiments should be double blind whenever possible. Okay, here's what I mean by that. What that means is neither the subjects, right, so the people receiving the treatments, nor those who measure the response variable know which treatment the subject received. Okay. Sometimes that second one is not possible, right? That's very difficult in some studies. So some experiments can only be single blind. Okay, so that would be when the subjects may know what treatment they're getting. So say you were doing an experiment with like types of exercise, right? The subjects can't be blind there because they have to know what exercise they're supposed to do in order to actually do it, right? But the people who they report their data to or who measure whatever that study wanted to measure maybe wouldn't know what type of exercise, what treatment each subject received, right? And again, the point of that is to try to eliminate bias, right? If you don't know which treatment is which, right, or which one you're receiving or which one you're measuring, then your natural biases are less likely to show up, okay? The one set of people that have to know which treatment each subject received is the people analyzing the data. So that's you, the statisticians. The statisticians have to know which treatment each subject received, okay? So that you can properly analyze the data, okay? And then the other term on here that you all may have heard before is the placebo effect, okay? So the statistical definition of that is it's the response to the dummy treatment. Okay, the dummy treatment is like the control treatment or the control group, right? So if, for example, you're participating in a medical study, if they want to put you in the control group, they're not just going to give you no medicine, right? They're going to give you a sugar pill that has none of the actual medicine in it, but you will go through the the motions of taking that medicine, right? And if you feel better, right, that probably is because of the placebo effect, right? Because you like think that you're taking a medicine, so then you feel better, right? Even though what you're actually taking is not medicine, but a sugar pill, right? Okay, so here on the screen, on the left side, I'm going to present you with a scenario. Okay, so we're talking about ultrasound and birth weight. Okay, um, there's pregnant women that were assigned to one of two groups. In the first group, they received an ultrasound. The second group did not. Okay, and then when the babies were born, they recorded birth weights. And the study found, or the experiment found, that women who received the ultrasound had heavier babies. Okay, so I have a couple questions for you here on, do we take the placebo effect into account? Like, is there a placebo really here at all? Okay, how could this possibly affect the response variable? Okay, was this a well-designed experiment? Was it double blind? And if it was, kind of identify that. If not, tell us why that's important. Okay, and then I want some suggestions on how you would improve the design of the experiment. Okay, so I'm going to pause the video here. I suggest you do the same and check back with me once you're done with this question. We'll see how you did. All right, y'all. So I've written my answers out here. Compare yours with mine, and let's see how you did. Okay. So in this experiment, the first thing that it asked was, does this experiment take placebo effect into account? I'm hoping that when you read this, you were like, wow, this is not a very well designed experiment. And you're right. It's really not designed very well. Okay. So the answer to the first question is no, because there wasn't really a placebo or a dummy treatment or a control here, because the group that didn't receive the ultrasound knew they didn't receive the ultrasound. So there was nothing to placebo there, right? For the mothers that did get the ultrasound, it's possible that seeing the image of their child would encourage them to maybe subconsciously to eat better and exercise more, which could result in healthier newborns, right? So that's, again, this idea of the confounding variable, right? That we wouldn't know if it was because of that extra, like, subconscious boost that the babies were healthier, or if it was actually because of the ultrasound machine. Okay. Number two said, was this experiment double blind, and why was that important? So, no, I talked about that a couple seconds ago. No. The people weighing the babies at birth probably didn't know whether that mother had an ultrasound or not, but the mothers did know the way that this experiment was designed, right? So at the moment, it's single blind, but not double blind. Okay. It's important because this could have affected the outcome, right? For a similar reason that I talked about in number one, right? When the subjects know which treatment they have, right, it can introduce some bias, whether that's conscious or unconscious bias. Okay, and the last part of this asks, how would you improve the design of the experiment? Okay, so really you need to make this experiment double blind. Okay, so you would have to treat all of the mothers as if they had an ultrasound, so that they wouldn't know which of the two treatments they received. Okay, so you would go through the motions of the ultrasound process, but for half of the mothers, you would turn the machine off, right? And then obviously for all of the mothers, you would have to turn the machine away from them, maybe put a blindfold on them, give them earplugs, something like that, so that they wouldn't know whether the machine was on or off. All right. Okay, last little bit here is I'm going to talk about statistical significance. Okay, so let's say you conduct an experiment and you go through this whole process and you get your data. Okay, the point of doing all of that is to see a difference in the response variable that wouldn't have simply happened by chance. Okay, if your experiment is well designed, right, you have the replication, the control, the random assignment, and and the comparison, right? If you have those 4 principles of experimental design, if your experiment is designed well, then your results, if there is a difference, it is likely that that difference didn't happen just by chance, right? Because of the natural variation of the universe, but actually happened because of the treatment that you imposed. So the term that we use for that is statistically significant. Okay, that's what we want. We want our results to be statistically significant. Okay, so the definition of statistically significant is that the observed effect, right? So, whatever happens in your experiment, the effect on the response variable is so large, right? Like so varied between the treatments that it would rarely occur just by chance. Okay. It's obviously possible that anything could occur just by chance, right? But we're saying this difference is so large that it would be very, very rare just because of the natural variation of the universe that this result would occur if we hadn't imposed our treatments upon it. Okay. Right. So, what this indicates, it indicates that our treatment likely caused the change. Okay. We're going to get a lot more into statistical significance throughout the year, but this is really the goal, right? Remember, this class is about a search for the truth, right? So when we conduct these experiments, right, we do these studies and we conduct these experiments, the goal is to identify a truth about a group of people, a group of things, a population of some kind, right? If that difference or that truth could have occurred just by random chance, then maybe it's not really a truth about that population, right? But if there is a big enough difference in those treatments or in the results because of those treatments, right? We say that the effect is too big, right? Too different, too much to have just been caused by random chance, right? By the natural variation of the universe. So it's likely that our treatment caused that change, and that change represents a truth about that population. If you can extend your results to the population. Obviously, any statistically significant results will obviously apply to the sample itself, right? It's going to give you a truth about that small group, that sample, right? In the next video, I'm going to talk to you a little bit about inference, which is how we can take those small truths that we learn through doing these experiments about samples and extrapolate them essentially, or apply them to our whole population, right? Because that's the real truth that we're seeking as statisticians, right? I know this video was pretty long. The next one hopefully will be a little bit shorter. All right, so I will see you all in the next video. Bye.